The process of formation of a mature female gamete is called oogenesis, which is markedly different from spermatogenesis. Oogenesis is initiated during the embryonic development stage when a couple of million gamete mother cell oogonia are formed within each fetal ovary. Oogenesis is initiated during the embryonic development stage when a couple of million gamete mother cell oogonia are formed within each fetal ovary. No more oogonia are formed and added after birth. These cells start division and enter into the prophase 1. No more oogonia are formed and added after birth. These cells start division and enter into prophase 1 of the meiotic division and get temporarily arrested at the stage called primary oocyte. Each primary oocyte then gets surrounded by a layer of granulosa cell and is called primary follicle. A large number of these follicles degenerate during the phase from birth to puberty. Therefore, at puberty, only 60,000 to 80,000 primary follicles are left in each ovary. The primary follicles get surrounded by more layer of granulosa cell and a new theca are called secondary follicle. The secondary follicle soon transform into a tertiary follicle, which is characterized by a fluid-filled cavity called antrum. The theca layer is organized into an inner theca interna and outer theca externa. It is important to draw your attention that it is at the stage the primary oocyte within the tertiary follicle grow in size and complete its first division, meiotic division. It is an unequal division resulting in formation of a large haploid secondary oocyte and a tiny first polar body. The secondary oocytes retains bulk of the nutrients-rich cytoplasm of the primary oocytes. At present, we are not very certain about this. The tertiary follicle further changes into the mature follicle or Graafian follicle. The secondary oocyte forms the new membrane called zona pellucida. Around it, the Graafian follicle is now ruptured to release the secondary oocyte ovum from the ovary by the process called ovulation.